schedule as well. So first we have our first year scholars, and the first one up is Julia Basso. And I can see your screen, Julia.
Okay, awesome. So my name is Dr. Julia Basso, and I'm an assistant professor in the Department of Human Nutrition, Foods, and Exercise at Virginia Tech, and the director of the Embodied Brain Lab. And my iTHRIV project is entitled Examining the Clinical Utility of Dance to Support Social Skills and Behavioral and Neural Synchrony in Individuals with Autism Spectrum Disorder. So autism is a neurodevelopmental spectrum disorder characterized by impairments in executive function, sensory processing, repetitive behaviors, and most prominently, issues with social skills and interpersonal communication. And we recently published a paper putting forth the synchronicity hypothesis of dance, which states that dance enhances neural synchrony in brain regions supporting 7 neurobehavioral areas that you can see here, which very much translate to a lot of these issues with autism spectrum disorder. And so when we engage in dance with others, we hypothesize that brain dynamics between individuals become synchronized. And so this paper and this grant here comes directly from this synchronicity hypothesis. And so we will test here that in both neurotypical individuals and individuals with autism spectrum disorder, dance will enhance social skills, or this interpersonal communication, behavioral synchrony, and neural synchrony. And so we're doing this through self-reported metrics, as well as physiological measurements at the level of the body, as well as measuring neural oscillations through electroencephalography. So tapping into the mind, body, and brain connection. Thank you.
Awesome. Thanks for sharing. Next up, we have Leanne Johnson. And
there we go. We ready?
Yeah, go for it.
My name is Leanne Johnson. What we know right now is that people that live in rural areas have more comorbidities than people that live in urban areas. But what we don't yet understand is for people with lung cancer, how these comorbidities influence their treatment and survival outcomes. So the overall track of my research is to improve personalized medicine within cancer care. For example, I really want to develop algorithms that will take patient information, including their comorbidity information, to help patients and providers have honest conversations about what care options are the best for an individual. So the next step in this process is my iTHRIV project. I plan to use data science techniques and a sample of data from the electronic health records at the University of Virginia. The first thing I plan to do is investigate all the types of comorbidities and their prevalence and patterns within a sample of individuals with lung cancer. Next, I plan to use the Charlson Comorbidity Index and the NCI Comorbidity Index to create comorbidity classes, similar to what we have right now in heart failure. And finally, I plan to analyze treatment and survival outcomes according to each of these classes. Thank you.
Thanks, Leanne. Next up, we have Gregory Madden. I can see your screen.
Hi, Greg Madden, Division of Infectious Diseases. With the SARS-CoV-2 pandemic, we noticed that MRSA rates in the hospital dropped by almost 1/3 during the pandemic, and it was unclear exactly why. MRSA is transmitted via contact by healthcare workers, and we know this in part due to social networks. And so our question was, how did social networks change during the pandemic? Using time-stamped EMR data, we constructed a network of patient and healthcare worker contacts. We divided this temporal social network with over 9,000 connections per day, comparing 10 months pre and 10 months post-pandemic. We hypothesized that the pandemic would favor reduced transmission due to cohorting, but instead we saw the opposite. And network modularity and density both suggested a lower threshold for transmission. And so we concluded that social networks did not help reduce MRSA, and may have in fact dampened the positive effects of COVID infection control measures. Masks may be more important for MRSA than previously recognized, and analyses to determine other factors responsible for this are underway. Thank you.
Thanks so much, Greg. So now up we have the second year scholars. So they will have 3 minutes to give their presentation, and we have roughly a minute, which is probably about one question, time permitting. If there is a question, I can look for it in chat, and I can also look for it on the raised hands. So first up, we have Kara Wiseman.
Thank you. I'm Kara Wiseman, and I'm an assistant professor in the Department of Public Health Sciences at UVA. So I'm going to talk a little bit about the work I've been doing as part of my iTHRIV Scholar time. I want to give a little bit of background, since we have more time, about my area of research focus. And so thanks to Dr. Ofili for setting up part of my slide by reminding us all that chronic disease remains a prevalent and important public health issue, and that tobacco use in particular remains a leading cause of preventable death in the US. And while tobacco rates have decreased over time, there still remain about 34 million adults who smoke in the US, about 13% of the population. And so there is a need for innovation to continue to support smoking cessation, both in the US and also globally. In particular, I'm interested in a mode of intervention delivery that's called mobile health, or mHealth. And mHealth is the use of technology to remotely track, monitor, or respond to or deliver an intervention for health-related events. We saw an example of mobile health intervention during our last talk. Using mobile health, we can take resources that are effective in person and move them to a more accessible mobile platform. Within the context of cessation, we have some examples such as mobile optimized websites, quit lines or telephone interventions, text messaging, smartphone applications, and even remote sensors. So for example, efficacious cessation interventions that include counseling or combination pharmacotherapy are really effective, but not everyone has access to those clinical-based services. So a mobile health tool can help bridge a gap in access. They can also serve as additional intervention components on top of efficacious interventions that are currently available. And importantly, 97% of US adults have a cell phone, and most of those are smartphones. So we have a real opportunity to reach a large proportion of the population. So my overall goal as a researcher is to develop effective and scalable interventions for cancer prevention and control. I just want to highlight some of the projects I'm currently working on that help me work towards that goal. So the first is a smartphone intervention study where we wanted to understand the impact of incentivizing self-monitoring on smoking cessation outcomes. The next one is establishing the effectiveness of publicly available smoking cessation resources. This is my iTHRIV project. And this one is related to the fact that we have some publicly available cessation programs, but it's a one-size-fits-all type of program. And we're not sure if that kind of program works equally well across different subsets of the smoking population. And so one case example is rurality and rural smokers who have higher rates of smoking and lower rates of smoking cessation attempts. So the goal of this study is to recruit smokers from rural and non-rural counties of Virginia and evaluate cessation outcomes if everyone's using the same text messaging cessation program. And lastly, I'm working on, I've submitted an R01 to look at longitudinal patterns of smoking cessation readiness and motivation to quit and quit attempts among tobacco using cancer survivors with the ultimate goal of developing new mobile health tools for this important population. So, thank you.
Awesome. Does anyone have a question? Really amazing and needed work. Next up, we have Sora Shin. And I can see your screen.
Okay, so my research programs, just one moment, it's hard. Okay. Yes. So my iTHRIV research program mainly focused on the abnormal eating habits, like the emotional overeating behavior, which has been developed after the life challenging, such as early life traumas. So stress experience like physical abuse and emotional neglect that occur during early developmental stage seriously impact our health outcome in later life. It could change the gene expressions, brain structures, the neuronal functions at the brain circuit level. So eventually, the early life stress produce several problems in our behaviors and cause mental issues. Particularly, the previous study indicate the strong association between early life stress and abnormal eating habits, such as binge eating behavior toward high palatable food, which I want to highlight today. So my lab is basically interested in how the early life stress cause the abnormal eating habits at the hormonal, molecular, cellular, and brain network levels. Previous studies suggest the stress experience activate neuroendocrine systems such as HPA axis and increase the corticosteroids that could impact other feeding-related hormone systems such as leptin, ghrelin, and insulin. But still, there are a number of questions that need to be addressed. For example, we don't know which hormone system is important for early life trauma-induced binge eating behaviors. And more importantly, those hormones are produced from peripheral organ, but its receptors are located in the brain, but we don't know how the receptor expressing neuronal functions has been changed after early life trauma. And lastly, we are curious to know the specific circuit pathway underlying this whole process. So these are the questions I want to address in my iTHRIV Scholar programs. And to answer those questions, we are mainly focused on leptin receptor expressing lateral hypothalamic neurons because our previous study shows the endogenous activity of this neural population is very responsible to the binge-like eating behavior of mouse exposed to early life trauma. We also found this neural population anatomically connect with multiple downstream targets and represent two distinct populations projecting to either the ventrolateral PAG and medial preoptic area. So we want to address how these two discrete circuit pathways make different contributions to binge eating behavior in early life trauma exposed animal. To study that, we manipulate each neural population using method named chemogenetics. We found the inhibition of ventrolateral PAG projecting neurons normalized the sustained binge-like eating behavior in ELT, the early life trauma exposed animals, in response to re-exposure of high palatable food, suggesting that this circuit activity is critical to reduce emotional overeating, particularly induced by early life trauma. So based on that, we are currently trying to understand how the leptin signaling can also contribute to this behavioral change and neural activity with circuit-specific manner. Thank you.
Thank you so much. Any questions? All right. Next up, we have Alex DiFeliceantonio. And I can see your screen.
Great. I am Alex DiFeliceantonio. I'm an assistant professor at the Fralin Biomedical Research Institute here at Virginia Tech. And I'm just going to briefly introduce the lab and my iTHRIV project today, which is focused around the fidelity of the gut-brain axis. And so what on earth do I mean by that? What I'm really talking about are signals that are derived from the gut that can guide and change behavior. And so we've known about these signals actually for a while. In 1942, Adolf manipulated the energy density of rat chow by mixing in cellulose, and he found that rats changed their eating behavior accordingly. And he concluded that rats eat for calories and provided some of the really early evidence that a nutrient signal from the gut must reach the brain and change behavior. The behavior of these animals, though, really stands in stark contrast to those exposed to so-called cafeteria diets, meaning diets composed of commercially available processed foods. In study after study, these animals rapidly gain weight compared to controls on a standard laboratory diet. And accordingly, the rise in processed food consumption has paralleled the rise in rates of obesity as well as type 2 diabetes. So this really indicates to me that modern foods, modern processed foods, must override brain systems that govern intake regulation and drive intake above physiological need. And so the lab is really interested in figuring out what is it about these foods that heighten their value and that lead to overconsumption. And so the long-term goal is really to identify these properties, which will allow us to design proper guidelines and interventions to improve quality of life and perhaps prevent some of the 300,000 preventable deaths a year that are attributable to dietary causes. And so we have some evidence of these different factors that influence the overvaluation of processed foods. We find that modern processed foods that contain combinations of macronutrients that activate different peripheral pathways are in fact overvalued. People will pay more money for them, calorie for calorie. And this overvaluation corresponds to differential encoding in the brain. My iTHRIV project focuses on another attribute of processed foods, is that they are rapidly digestible. And here we are testing if nutrient availability or the speed at which those nutrients can signal can change behavior. And we have some preliminary evidence where we've manipulated how fast a carbohydrate can be digested, showing that the fast digestible carbohydrate actually does change behavior. People like it more, even though we gave them the same amount of calories. And so with that, I'll take questions and say thank you.
Thanks so much. Any questions for Alex? All right, next we have Laurie Brenner.
I think we have a question, Jessica.
Oh, I'm sorry.
Yeah, that's fine. I had a
Oh,
quick
I'm
question.
sorry. Dr. Feely.
Yes, thank you. So these are human studies?
Yes.
Okay. Fascinating. Great.
Thank you. Yeah, I should have been clearer. I started with the rodent work, but the two studies I presented are done in human participants.
Okay, wonderful.
All right, Laurie Brenner, thank you. And I can see your slide.
Hi, everyone. My name is Laurie Brenner. I'm an assistant professor in the Department of Neurology at the University of Virginia. And my research interest is the intersection of autism and epilepsy. Specifically, I'm interested in brain mechanisms that may explain the high rate of overlap between these conditions. So the prevalence of epilepsy in autism is around 12%, with some studies reporting prevalence as high as 30% compared to a 1% prevalence in the general, 1% epilepsy prevalence in the general population. And so the central hypothesis of my research is that autism plus epilepsy is a phenotypically distinct subtype on the autism spectrum that is associated with frontotemporal network disruption. And I used a retrospective chart review for my iTHRIV project to identify 395 individuals at UVA between the ages of 2 and 25 who have the dual diagnosis. And for this pilot study, I only included individuals who had undergone comprehensive neuropsychological testing. So this gave me a sample of 31 for this initial phase of the project. And what this allowed me to do is implement a very rigorous review process for the epilepsy and autism diagnoses to confirm those diagnoses. I ended up with a sample of 23 who met rigorous criteria for both diagnoses. And so 78% of the sample had an abnormal brain MRI, as shown here in figure 1. An abnormal brain MRI was associated with lower intelligence. In fact, IQs were almost 30 points lower on average in the group with an abnormal brain MRI. EEG, MRI, and seizure semiology data were reviewed to determine epilepsy type and seizure onset zone. And for patients with focal epilepsy, experts rated the likelihood that seizures were arising from the left versus right hemisphere and from frontal, parietal, occipital, or central regions. So in this sample, focal epilepsy was more common than generalized epilepsy. For individuals with focal epilepsy, frontal and temporal regions were identified as the most likely source of the patient's seizures. These data are shown in figure 2. So you can see the likelihood ratings are on the y-axis, And then lateralization and localization are on the x-axis. White bars represent the left hemisphere, blue bars the right hemisphere, and the data are further subdivided by frontal, central, temporal, parietal, and occipital regions. And so we conclude that an abnormal brain MRI may be one of the defining characteristics of autism plus epilepsy. In the autism population more generally, pathological findings on clinical MRI are rare. And so although the sample is small, we also have preliminary evidence that focal epilepsy may be the most common type of epilepsy, which is concordant with data from 2 other published studies. And so in order to address some limitations, including the small sample size and answer additional questions about brain characteristics in this population, further study is currently underway with the EEG data from the larger sample of 395. And future directions also include analysis of the MRI data, possibly using a lesion overlap mapping approach to identify regions of interest where there may be overlap in pathology. So I want to thank my mentoring team and NeuroNext, iTHRIV, and STAR for supporting my research. Thanks for your attention.
Great job. Dr. O'Feely left a question in the chat about what program for building your poster.
About what now?
What program you used to oh, I'm sorry.
Oh.
Or is that?
Yeah, I like the Build poster. It was pretty nice. I was just asking, what's the software that does that?
I just used PowerPoint for that with animations.
Oh, cool. All right.
Yeah.
Thank you.
Not too fancy, but thank you for the, I'll take that as a compliment. Thank you.
It is a compliment and definitely great research. Thank you.
Thank you so much.
Okay, so next up we have our last cohort, which are the Scholar alums. So they have 5 minutes to talk about their work at iTHRIV and time for questions after that. So first up we have Jeff Sturek.
Can you see my screen?
I can. It's a little bit, so I'm only seeing like the right side of it. Is that for everyone? Okay, perfect. Yeah, you're good right there.
Thanks for emceeing, and I get to slow down a little and take 5 minutes to tell you about how I spent my time as part of iTHRIV. And if you take anything away from it, it's that key in anything for this early career phase is having great mentors, mentorship team, and colleagues, and that's certainly been true for me as part of iTHRIV. And so I want to just give you a broad overview of the focus of my lab, which I'm calling translational pulmonary immunology. And I really like to be at the interface of sort of multiple parts of this. I'm a pulmonary and critical care doc at UVA. And so all of my focus is around lung inflammation and fibrosis. And we use clinical data, with patient samples that we can collect. We want to do high dimensional analysis of this. Between my time in my PhD and in my fellowship, one of the things I was struck by is just the explosion of these high dimensional techniques. And so one that we use is called mass cytometry that I'll talk a little bit about to get as much data as possible out of these patient samples. And then we take things into mirroring models, looking at sort of basic disease mechanisms so that we can understand this spectrum from health and disease and how we can drive disease states towards a more healthy state. And so some examples of different projects that we focus on are this particular type of B cell, which are called B1 cells, and these antibodies that they make. And I'm going to tell you more about this in detail in the next couple slides. We have mirroring models of this, and we have patient samples looking at this kind of stuff. I had the luxury of having our cohort had COVID happen in the middle of our first year, but really for me, that ended up being a huge opportunity to be involved in lots of different things that I never would have predicted. So I was involved in a convalescent plasma trial here at UVA, which ended up being funded as a supplement through the iTHRIV grant, which I am, forever indebted to our iTHRIV team for making that happen. And through that, I also helped establish this thing we're calling the Joint Emerging Diseases Initiative, or JEDI, which is a collaboration with Patrick Jackson and ID, where we do other clinical trial work with COVID. We have a translational project with this mass cytometry or high dimensional immunophenotyping in COVID-19, and there's an excellent postdoc in my lab, Riley Annan, who is pioneering that. developed this 70-plex barcoding assay where we can measure 70 different patient samples at once. And then we have some of this same B cell work going on in COVID. And then I also see patients who end up with lung problems related to cancer. And so I'm not going to talk about those, but there's ongoing projects in that space as well. But about these cells that I think are really cool. So unlike the normal B cells that you may have been reading about that are making all the antibodies after you've gotten your vaccine, these B1 cells are actually sort of innate B cell subset. They're present from birth. And they are evolutionarily conserved, and they make these antibodies that are evolutionarily conserved and don't require help from the other side of the immune system to make antibody. These antibodies, which are referred to as natural antibodies, recognize multiple epitopes. So unlike these highly specific switched antibodies from what are conventionally called B2 cells, these recognize lots of different things. They recognize self-antigen and foreign antigens as well. And what we think they do is provide this early innate response to infection, and that they mask autoantigens and so sort of prevent detrimental inflammation. And the other interesting thing is that, to me, is that they're located around the lung in this sort of lining of the lung and the pleural cavity. And this is just a schematic of what these different epitopes are. So basically, altered self or microbial antigens are all recognized by these similar receptors. And so what we do is we look at these in the lung. And so we have mice that are genetically modified, so they have increased B1 cells. We quantify their lung damage and fibrosis. You can actually put a mouse in a CT scanner. You can quantify that. We take the lungs and look at fibrosis and histology. We do flow cytometry and look at the different immune cells. We measure the secretion of these antibodies. And the summary of this work is that what we think is going on is that there's this lung injury signal, which talks to the pleural B cell, and that this B1 cell then traffics to the lung where it makes these IgM against oxidation-specific epitopes, and that those inhibit fibrosis. And so ongoing work is now we're extending this into patient samples, and Eva Otopolova is this excellent pulmonary critical care fellow who is a new iTHRIV scholar. Shout out to Sana for establishing that, which is awesome. We're looking at how these cells traffic by basically putting in these little mouse chest tubes and seeing where the cells go. This just shows we can follow them. But also we are looking at this in COVID as well, and just fast forward through to say, which is we've got some data from humans that there's changes in B cells and natural antibodies. We've used the mouse model that's established here. We helped establish at UVA to look at these different chemokine receptors and trafficking of B cells in those. And I also had the opportunity to submit this project on a PPG, which is a really cool opportunity as an early stage investigator. So hopefully that will not get completely trashed when it gets reviewed at the end of this month. But I should spend most of the time on this slide because I've had the privilege of being part of Colleen McNamara's lab, and just all those people are outstanding people. And if you have the opportunity to work with them, I highly recommend it. And the same goes for the iTHRIV team and all my other collaborators. And so with that, I'll take any questions.
Thanks so much, Jeff. Any questions? Really impactful work. Next we have Kelly Shaffer. I can see your screen.
Great. All right, so I'm Kelly Shaffer. I'm an assistant professor here at the University of Virginia in the Department of Psychiatry and Neurobehavioral Sciences in the Center for Behavioral Health and Technology. So I'll share a little bit about my iTHRIV Scholar project, optimizing efficiency and impact of internet-delivered insomnia treatment for caregivers. So sleep disturbance is a really common problem among the about 1 in 6 of us who provide care to a loved one to help them take care of themselves when they're sick or disabled. So in the general population, at any given time, about 1/3 to 1/2 of us say that we're experiencing sleep disturbance. In the caregiving population, that's up to about 90% of caregivers report clinically significant sleep disturbance. So in good news, our center has developed a online fully automated cognitive behavioral therapy program over the past 15 years or so that has worked really well to treat insomnia in the general population. And we think that the online delivery will work really well for family caregivers whose responsibilities and sort of everything going on in their lives makes it really difficult for them to access sort of traditional in-person care. So the not as good news is early results from our most recent trial of shut-eye among older adults shows that the individuals participating in the trial who self-identified as family caregivers didn't report as great of a benefit from the program as non-caregivers did. So we are following up on those results in my iTHRIV study, which is a single group open label trial of shut-eye among family cancer caregivers who are experiencing sleep disturbance. And so I want to give you just a little teeny tiny slice of results from the first 3 participants who went through the study. And the goal of these findings are really to help us understand what needs to be potentially changed about shut-eye for it to work the best among family caregivers. And also hopefully shed some information on how we can make sort of other existing internet interventions more accessible to caregivers as well. So I'm going to show you, again, 3 different participants and their feedback on the client satisfaction questionnaire, 3 items from that questionnaire. And these were all spousal caregivers, but they really differed in terms of the amount of care, in terms of time providing care to their husbands, and how many cores they completed out of 6 total in the shut-eye program. So the first question is, to what extent did shut-eye meet your needs related to sleep? And our first 2 participants said that really most of their needs had been met and cited that they really like some of the standard cognitive behavioral therapy techniques that work really well in the general population. Our third participant had a lot to say. And she really indicated that she really wanted more tailoring for this program to work for her as a family caregiver. The next question, in an overall general sense, how satisfied are you with shut-eye? Again, our first 2 participants saying that they sort of were really pretty satisfied. Unfortunately, again, that third participant saying that she was not as satisfied and really would have preferred some tailoring. And then lastly, again, would they have used shut-eye again? Our first 2 participants saying yes. Probably they would use that sort of general non-tailored shut-eye program again, while the third participant indicated that she would probably look for something that was more tailored for the caregiving experience. So this data has given us a lot to think about in terms of what kinds of tailoring, what level of tailoring is necessary and sufficient to make sure that caregivers can optimally engage and benefit from our interventions. Because on one hand, we know that this is a really, this group has a lot of psychosocial concerns that need to be addressed now. And we have a lot of programs that we think could work well. As our first 2 participants showed, probably some caregivers can gain a lot of benefit from the programs that we already know work. On the flip side, we don't want to be trying to fit a square peg in a round hole. As our third participant showed, there's probably some caregivers who need some specific content to address their sort of unique problems related to sleep and caregiving, and to make sure that they're feeling more seen as they're going through these interventions. So in good news, we were awarded an R21 from NCATS to answer this in a little bit more detail, and we're getting that study up and running now. And so stay tuned for more results. And I would just want to thank everybody who has really supported me particularly to the iTHRIV program for providing really career-changing opportunities, as well as everybody who made my participation in the program and work on this study possible. So please be in touch with any questions, and I'm happy to answer one now.
Thanks so much, Kelly. Just looking for any questions.
A quick question, kind of regarding what that third participant was saying. In that main study, did you have information about, why it wasn't effective, thinking about people who are being woken or their sleep is disturbed because of the person in their care who, might need medication in the night? And at that point, what can be done to improve caregiver sleep if they have to wake up to care for someone else?
That's a great question. That's exactly what that the Bessie study and now our 21 study is looking at. We asked them sort of what's going on with them in the middle of the night. And I would say that it wasn't that the program wasn't effective. Actually, that particular participant's insomnia severity index went down quite a bit. But she was looking for something that was more caregiver specific. And so we do have plenty of data on that, and we'll be answering that question with further data analyses and with the R21 study. So thank you so much.
Congrats on the funding.
So last but not least, we have Tanya Evans, and I can see your slide great. And Tanya, you're muted, I'm sorry. Tanya, oh, there you are.
Sorry about that.
That's okay.
My name is Tanya Evans, and I'm an assistant professor at UVA in the School of Education and Human Development. And today I'm going to talk about whether or not parent-child brain synchrony can provide a physiological marker for individual differences in social skills. And first of all, clearly Julia and I need to talk because our projects are very, very related to each other in terms of looking at neurosynchrony and ASD. So EEG brain synchrony. We use mobile EEG headsets to look at whether or not there's synchronous brain activity between parents and children. So an exemplar of low brain synchrony is in the bottom, and an exemplar of high brain synchrony is in the top. And there's evidence for parent-child brain synchrony from the literature where they look at both direct and indirect eye gaze between an adult and an infant and show higher brain synchrony during eye contact, which is really interesting. So in terms of social engagement and autism, there's social communicative differences, which are a core feature of autism, and this includes gaze aversion and atypical nonverbal communication. We know that their interactions are less synchronous, and this is really important because there's a reciprocal nature of communication where in non-autistic samples, synchrony is associated with rapport. So we're interested in how well automated measures of behavioral synchrony correspond to qualitative ratings of engagement in parent-child dyads. A little bit more about social engagement and autism. Autism traits are on a spectrum where there's a wider difference in autism traits that may lead to a wider disjuncture in reciprocity. So we're also interested in how parent and child autism traits interact to predict their interactional synchrony. The purpose of this study, we're really focused on how traditional neuroimaging studies focus on the observation of autistic individuals in isolation, where here we're quantifying social engagement between autistic children and their parents by using simultaneous EEG recordings and integration of multiple streams of data to measure brain synchrony. We have parent-child dyads ranging in age from 6 to 11 years old with and without autism. And using electroencephalography, we record brain synchrony between parents and children during various tests while they're fitted with these 20-channel mobile EEG headsets. So we use an analysis called motion energy analysis. And this is a method for quantifying the amount of movement in video data where the number of pixels which change in a given region are quantified between two time frames. It's a crude measurement of movement, essentially. And the algorithm that we use segments two coupled time series into moments of symmetry and moments of non-symmetry, where a time point is symmetric if it is related to nearby time points. And the similarity between time points is maintained across time. We look at prospective and retrospective similarity patterns. And here's some example data here where we look at a physical synchrony during musical free play versus a musical task, where we have a free play condition on the left and a task condition on the right. And as you can see, there's a higher correspondence of physical synchrony during the musical task condition. Now to move on to the EEG data, we look at parent-child brain synchrony across an interval of 4 seconds here. And we see that there's higher brain synchrony for child-led synchrony. So when we look at a lag, we're able to induce that the child was leading the synchronous patterns. So this is just some sample data. We're in the midst of collecting more data now, and so stay tuned. Just acknowledging all of my mentors and collaborators here at UVA, as well as the funding sources. Thank you. I'd be happy to entertain any questions now.
Thanks so much, Tanya. Any questions?
I'll ask a question if nobody. Hi,
Sure.
Tanya. Good to
Hi.
see you.
How
Sort
are
of.
you?
You're like
Yeah.
very much in, like you're very dark. Your face is.
Sorry.
Yeah. So I'm sorry. Can you clarify? Did you actually do analyses on the MEA yet? Like any movement analysis, or you did not? Those are these. You're still looking at the results for the movement synchrony.
This
and
is
then relate it.
This is
Yeah.
the MEA for the physical synchrony. This is like some pilot data here.
Okay, but you don't have any group results yet on this?
No.
And can you, like, is there a reason, because as you said, MEA is a very crude measure.
Yep.
And I'm trying to wrap my head around what that might mean. Like it could also mean like a lot, it's, even lots of movement, right?
Right, right.
So I'm curious to know if you've considered other ways of looking at movement synchrony, like open pose or some other,
We haven't, but that's a really good suggestion.
Yeah. Well, I don't know. I mean, I'm like MEA is like this kind of,
Gold standard a little bit.
well, yeah. And then there's a lot of findings. I'm just always having a hard time interpreting what because it's not tradition. It's not synchrony in the sense of like you do the same thing, right? It's more like,
Right.
oh, I move a lot, you move a lot, and we happen to do
Right.
this at the same time or in a certain, time frame. So
I think
anyway.
if we see correspondence across the MEA analysis and the EEG analysis, then we could be a little bit more confident in the interpretation of the results.
Sure. Yeah, I could see that.
Yeah.
That's a good point. All right.
Yeah.
Thank you. Very exciting.
no problem.
Excited
Thanks
to
so
see
much.
the findings.
Thanks for coming.
Wonderful job to everyone. It's just amazing to see the new ideas from the folks starting off and the growth throughout the cohorts. Thanks so much for your time and effort. Just a reminder that we have some closing remarks, so you can leave this breakout, but don't leave the full meeting. We'll see you back in the main room. Thanks so much, everyone.